Good morning, everyone. I'm very happy to be here and feel excited. And I want to first of all take the opportunity to just thank the Prize for Life and also the Dream Challenge for just organizing and hosting this challenge and also making it happen. And my name is Guang Li, and I'm with my colleague Liuxia Wang here. And we are from the company called Sentrana. So just to take another opportunity to just make some advertisement about our company. Actually, we are based in Washington, D.C., and we are a scientific marketing company that we use a lot of quantitative approach, such as mathematics, modeling, statistics, machine learning, and optimization to help our clients to solve their business problems in marketing domain, such as how to find the optimal price for certain products and how to figure out what is the best selling opportunity for certain customers. So today my topic is going to be pretty straightforward. So first of all, I'm going to talk about the problem and its challenges. And I think I will go through very quickly for this one because I think you guys have a lot of context about the problem itself. And then we just introduce the kind of like the techniques for us to process the data, do the exploration analysis. And also I'm going to introduce the structure of a model and as well as the results and conclusions we have. And for the last part, we think that is probably be very, very kind of like interesting to talk about some implications we have from our model. So for the first part, the problem itself is actually pretty straightforward. It's like to use the data from the first 3 months to predict the future progression rate of ALS in the month 3 to month 12. And I want to just talk a little bit about the challenges here because that really just make me suffer a lot during the competition. So first of all, there's no proven causal relationship and factors for this type of disease, which also makes this challenge interesting, but it's also painful. And the data is actually very dirty because there is a lot of missing data for a lot of different variables, and there are a lot of, the scale is different, there is a lot different measurement for different variables, and also there are so many data. I know that Prize for Life wants to give us as many data as possible. I thank for that, but that really make our lives really hard. So we need to figure out a kind of like a good techniques for the variable selection to figure out, okay, this is the best informative variable that we're going to use, rather than just put everything into our model. So actually our goals is actually twofold. So first of all, we want to discover if there are any interesting variables or interactions of these variables that can be used as a future kind of like biomarkers for the ALS progression. And second one is like we try as hard as possible to just design an algorithm with a better accuracy. So the first part for us is, first step is trying to figure out what is the right data for use as input of the model. So this decision is actually based on two criteria. So first of all, we do some literature review. It has been shown that in a lot of research, some kind of factors has already been identified in both significant and effective to predict the future ALS progression. For example, the location for the onset, the age, and some lab tests, for example, the uric acid value, and also the time difference between the onset and the diagnosis. And second criteria is actually we take a look very intensively at the data and trying to figure out, okay, this is the data we want to use. So the first very intuitive thing comes to our mind is actually, since we use the historical data to predict the future, then it must be very informative for, we use the first 3 months data. So we found that for a lot of patient cases, actually, the ALS FRS score is actually consistent with their future progression. But the bad thing that for a lot of times, there are a lot of noises for the first 3 months data. There are a lot of fluctuations and variations. So we want to consider both together. So the way we do that is actually, first of all, in order to take this information, we just fit a 9 for the first 3 months ALS FRS score and use that slope as a predictor. And also we define an MSE, the mean squared error, to represent the credibility of this score. And second thing that is actually pretty interesting, because for a lot of research that has done already, people tend to just look at this score as a whole. I mean, look at the total score. But we'd rather to think, okay, why not just to take a more granular level, which we actually, first of all, we look at the 10 questions, because if you look at these 10 questions, you'll find that actually it represents the different functionality for different body parts. So for example, the first 3 questions, speech, salivation, and swallowing, is actually represent, how your face muscle works. So based on this, and also we have the metrics for the correlation for these 10 questions for this score. And we find that actually we can divide them into 5 groups, such as face, hand, body, back, and chest. And there are some interesting things here. So first of all, you'll see that the hand and body, actually, these 2 parts are correlated with each other very much. But actually for the face and the chest, the correlation value with other body parts is actually relatively small. So we think probably we can just treat these 5 different parts differently and just put their score and slope into our model. And another interesting thing that we actually we look at the initial health condition for each patient, and we find that there are actually some nonlinear relationship. We just fit a very simple quadratic model for between the slope, the future slope, and the ALS-FRS score for the last trial at the first 3 months. And we can see that there is actually the first order and second order term, they are both very, very significant. And if you look at this curve, you can see that, okay, when the score is larger than 26, actually it is positively correlated with each other. But when it is smaller than 26, it's actually negatively correlated with each other. So this is kind of very interesting, because the possible explanation can be, if people already have a high score, which represents a relatively healthy state, or have already come to a bad situation, probably that means they've already come to some stable kind of state, that the future degeneration of the health condition will be kind of like slow compared to others. And also, I think another very interesting factor we identify, I think another team also talked about, that is actually the time difference between the onset and the time we measure. So we also do some data transformation that if we assume that the patient health condition is pretty good when just get onset. So the total score should be the full marks, which is 40-0. And then if you just minus the first trial ALS-FRS score and divide it by the time difference, this is actually another source of historical information about the progression slope we can use. So actually this is just kind of like selected variables I'm talking about. Actually, we define a lot of variables. So first part is actually the information for the first 3 months ALS-FRS score information. So as you can see, we have the slope for 3 months. We have the slope for the onset. And we also kind of like measure the different body part differently. We define the score for each 5 group, and also we define the changing rate for 5 group. And except for that, we also have information such as onset information, and the family history, if any family member has diagnosed with ALS before, or the vital capacity information, and there are some kind of like the result from the lab test we can utilize, and also the vital signs such as the weight, the pulse, and the breathing rate. So in terms of handling the missing data, for the data that has a lot of missing values, we define a binary variable to indicate if this variable is missing or not. For the variable that has just kind of like small amount of missing data, we just impute using the mean or the mode of the data. So we propose to use a random forest algorithm. I guess a lot of you guys must very familiar with this algorithm, but I just want to talk a little bit about the overall kind of idea. So random forest is actually an ensemble classifier, and that it just generates a lot of different independent regression trees, just from the subset of the data, and then trying to aggregate the result as an output of the model. So, but we feel like it is probably not a good idea to just throw out our data into just one model because of the data characteristics we have. So if you look at the variables, for example, for like uric acid value and the time difference between the onset and diagnosis, they are actually as high as about 80% of the data are missing. So due to the kind of instance random forest work, because it just uses kind of like aggregate result and just voting for the best prediction it can have, that means these kind of two variables, their kind of like explanation power will be greatly diluted. Another thing is that actually there are some inconsistent measurements for different type of variables. For example, for the vital capacity, each patient either have a faster vital capacity or a slow vital capacity. But actually, it is if we just fit two different separate linear models for these two variables, you'll see that the coefficient is actually vastly different. So we don't want to put them together and just use a combined model. So we come up with the idea to maybe to take care of the different situations respectively. So we have this mixed model, we call it mixed model, and so there are actually 4 models. The base model is actually just based on all the data, and we use a binary indicator to indicate if it is missing or not. And for the submodel 1, we just develop it on the subset of data if the fast wider capacity data is available. And for the submodel 2, we actually depends on if the uric acid value is available or not. And for the submodel 3, it actually depend on the diagnosed delta data, which is the time difference between the onset and diagnosis, if it is available. And we do that. We just test it on the tested data, and it seems like compared to the base model, each submodel, just on that particular subset of data, they performs better in terms of the reduction of RMSD. So if you look at this number, you probably think, okay, yes, it does not matter, just a small number. But if you think about how close each solution with each other and how difficult for us to improve the accuracy, even like 1% is actually pretty significant for us. And this result is actually consistent when we do on the validation data. And also the logic for combining this model is actually pretty straightforward, because we first of all use a model that can improve the accuracy most. And then for the remaining data, we go to the second model that is relatively increase the accuracy a little bit, and then finally we come to the base model. So to summarize, I think first of all, we achieved the accuracy of our RMSD, which is 0.05112, compared to the baseline algorithm, which is 0.053. And because we do a lot of cross-validation for the variable selection, we successfully avoid the problem of overfitting that a lot of kind of like challenging participants they encounter. And also, we successfully identify a lot of very, very interesting relationships and dynamics and a lot of new variables. Maybe it's kind of like interesting for the future to study. And also, from the random forest point of view, it has a very inherent matrix to measure the variable importance. So we can see that from our model, the length of the time from onset, the deterioration rate from onset till now, the changing rate for the vital capacity, the ALS FRS score change rate, they are kind of like top most important variables. So if you look at this is actually the variable importance for the 4 models. You'll find that actually for all of them, they are consistent. But if you look at, for example, for the submodel 2, you'll see that the uric acid value just rank a little bit higher, because here we just take care of this one particularly. And it is also true for the submodel 3, which is, you can see the onset diagnosed delta difference is also the third most important. So what does that mean? We don't want to just have kind of like accuracy and just give to price for life. Okay, we'll go away and we'll take the money, So we think that from an outsider's point of view, who has not any experience in computational biology, bioinformatics, or ALS disease, probably we can, just from our model, there are some interesting implications. So first thing we think about is like, is there any different stages for this disease? Because first thing is that if you think about the most important variable actually are the time difference from the onset. That means it is not time independent. Second is like, if there is a stages for different patients, is there heterogeneity for the length of the kind of like stage for each patient? And what kind of factors actually contribute to this difference across these patients? So probably some kind of variables, it does not just affect this progression rate directly, they just through some certain structures, they affect the length of the stages, and then it will demonstrate different progression rates. And second thing that here we have seen this matrix again is that would be it more beneficial to look at the different body parts independent, kind of like individually rather than combine them together, because there are so many interesting correlation here. So is there any reasons that, you know, from the disease perspective of view, that actually why certain body part they are correlated with each other are more than other, you know, the body part? And also, I mean, just based on my naive kind of like intuition, like, is that possible that for certain neutrons that controls this body muscles, they're close with each other or far from each other, that will contribute to their correlation? And also, is there any transmission mechanism? Because the chest muscle is always, for most of the times, the last one to deteriorate. So I think there's a lot of things to do in the future. I think, yeah, that is end of the presentation. Thank you very much. So, does anyone have some questions? Seems people are just eager for lunch.
Yeah, everyone's really eager. I have some papers to send you because that's exactly one of the latest trends in the field is this correlation and distance between the neurons in the brain and the body parts.